Welcome back. This is part 2 of our lecture for lesson 1, Brain Basics. We'll be jumping right in where we left off. This time we'll be talking about neurons, neurotransmission, and how drugs can affect that process. So this is part 2 of a 2-part series on brain basics for lesson 1. So jumping in where we left off, we were talking about larger structures of the nervous system, the brain, the spinal cord, these different branched components of the nervous system. Now we're taking a dive more narrow into the building blocks of the nervous system. This is a picture of a nerve, a neuron, and it also includes a blown up image of the space between two neurons. So I'll be flipping back and forth with slides that have definitions of these components and this image here. So jumping in, again, like I said, the neuron itself is the most basic component of the nervous system. And it has at least sort of 3 broad parts. There's the dendrite, which is this branching structure that receives and transmits chemical messages between neurons. And then the axon, which is the long part of the neuron, which carries the message across. And so in this image here, and I will get my highlighter out so that you can see. So the entire structure here, this is the neuron. And the dendrite are these branching structures here. So these parts of the neuron that are branching off of the cell body. And they're responsible for sending messages between neurons. Then you have the axon, which is this long tube that transmits information within a single neuron. So the information and the signal moves down the neuron through the axon. Some other key terms related to neurons and neurotransmission. You have the action potential. So this is the another name for that electrical message that's being carried down the axon, the long tube that's carrying the message within a single neuron. And in order to facilitate that process or that transmission of the electrical signal is the myelin sheath. So this is a coating along the axon, which really kind of serves as insulation for the electrical message. So if you think about a neuron like a wire, we don't just have raw wire that connects our appliances to the outlet. They're wrapped in insulation or a coating in order to preserve the electrical current that's moving through it. Similarly, in the neuron, our myelin sheath is designed to help insulate and protect us, protect the signal as it's being transmitted down the axon. So this blue component here is the myelin. Let me get my highlighter going. This is the myelin here. And what it does essentially is it allows the signal to jump from these different nodes. So this would be one node. This would be another node. And it basically speeds up the transmission and keeps it contained within the axon itself. It's a very important function. The breaking down of myelin sheaths is something that's related to dementia as we age. And then at the end of an individual neuron is the axon terminal. So that's the part of the axon where neurotransmitters are stored and released. So it's the end of the individual neuron, and it's the part that precedes the connection to the next neuron. So the axon terminal butts up against the dendrites of the next neuron, and it the space between those two things forms a synaptic gap. So you can see here in this image, in this section of the image, you can see the synaptic gap, and you would have the presynaptic cell would be, let's say, this neuron right here that's sending information. Once it gets to the very end of the axon, it reaches the axon terminal, right? That's this very tip of the axon here. And then that would be this area. Then there's a space, and then the dendrite of the next neuron, which is down here, forms this space here. And this is where information is communicated between the cells. We'll talk about that in more detail momentarily. Located within the axon terminal are vesicles. So at the end of the axon, as the electrical signal is getting sent down the axon, it reaches the axon terminal where there are round structures that store and release neurotransmitters. So you can think about all information traveling within the brain as a neurochemical, chemical reaction. So there's an electric signal that's being sent down the axon, and then that triggers the release of particular chemicals, neurotransmitters, from one cell to the other that then sparks another electrical reaction inside the next neuron. The synaptic gap, or the synapse, is the space between the two neurons. I mentioned that a moment ago. And the neurotransmitters themselves are these chemical messengers that influence behavior in a variety of ways. So there are dozens of different types of neurotransmitters that have already been identified, and there are potentially even more. I mean, it's almost hard to imagine how many there could be, but there have been at least 30 or so identified. We'll be focusing on a particular subset of neurotransmitters. But what they are essentially the particular chemicals that send particular messages in between neurons. And this is where drugs can really impact the brain. So in addition to, I mentioned in the last lecture that some drugs can just kill neurons, right, in a certain brain area. So boom, just killing brain cells. Another way that drugs can impact the brain is through neurotransmitters. So either speeding up, slowing down, mimicking, blocking the performance of these neurotransmitters. On the other side of the synaptic gap are the receptors for the neurotransmitters. So when the electrical signal reaches the axon terminal, then the neurotransmitters are released into the synaptic gap. And then on the other side, the start of the next neuron, the dendrites, have receptor sites on them that receive information from the neuron before it. So I'm going to flip backwards to our picture here. So you can see this process that I just described. Where you have the vesicles at the axon terminal that are housing the neurotransmitters, which are these blue dots here, inside the vesicles, inside the axon terminal, here at the bottom of the axon. This space here is the synapse, the space between the axon terminal of one neuron and the dendrites of the next neuron. And then these blue components here are the receptors. And so these are located on the dendrite of the next cell, and they receive the neurotransmitter from the axon terminal that's being released from the first neuron. And then finally, another important term to be aware of when we're talking about drugs and the impact that they have on our nervous system is this idea of the blood-brain barrier. So it, substances enter our bloodstream, and through our blood, they circulate all throughout the body. And when they get to the brain, however, there is a particular layer of cells in between the blood vessels in the brain and the brain tissue itself. And that layer is called the blood-brain barrier. And what it does is it really regulates what from the blood is allowed to enter into the brain. And the idea is that it would prevent harmful substances from entering the brain. Um, the what makes drugs is that they do have an impact on our brain, that they are psychoactive, meaning they have some psychological and mental effects, and meaning also that they're affecting our brain. And so substances that transmit through the blood-brain barrier are going to be psychoactive, and they're going to have an impact on our behavior. How they do that, we'll talk about in a moment. But first, it's important to understand just the basic function of neurotransmission, and the transmission of signals between neurons across the synapse. So I talked about this briefly, but we'll go into a little bit more depth here. So as the nerve impulse, the electrical signal travels down the axon, it reaches the axon terminal. So it moves from the dendrites down into the axon terminal, and that's where the vesicles release the neurotransmitters into the synapse, and those transmitters move across the synapse into the dendrites of the next neuron. Those neurotransmitters carry information, and they spark the electrical signal in the next neuron. And it does that first by binding to the receptor site located on the dendrites. And this is a lock and key mechanism. So for every particular type of neurotransmitter, there is a very particular type of receptor on the other side of the synaptic gap on the dendrite. And those neurotransmitters only bind to those particular receptor sites. Once those receptor sites are full of neurotransmitters, anything that's left over free floating in the synaptic gap is broken down, or it's taken back up into the axon terminal, which is called reuptake. Then, once that chemical signal has reached the dendrite of the next neuron, that's when that triggers the electrical impulse traveling down the neuron, and it repeats all over again. This happens so incredibly quickly. I can't give you the exact millisecond, but it is almost instantaneous. So, as I mentioned, there are several different types of neurotransmitters, several dozen. We will be talking about 8 for our purposes that are very important in understanding how drugs affect human behavior. The first is acetylcholine. There are high concentrations of this neurotransmitter in the brainstem and the midbrain. This is involved in our autonomic nervous system and our muscle contractions. And so Botox, for example, affects this neurotransmitter by preventing its release into through the synaptic gap. It stops it from being released. which then stops the muscle contractions for where it's being administered. So Botox is actually a very effective treatment in terms of managing migraines because it stops muscle contractions in the face and head, which relieves tension that's associated with migraine headache. It's also the case that acetylcholine has been implicated in Alzheimer's disease, but that was a theory. And then when people developed drugs to affect acetylcholine in the hopes of alleviating Alzheimer's symptoms, the medication isn't very effective. And so many people are starting to second guess whether or not that neurotransmitter is actually causal with Alzheimer's disease. Another neurotransmitter is norepinephrine. This is a transmitter that's located throughout the brain and the body. This is a neurotransmitter that's responsible for our sympathetic arousal. And so you'll remember parasympathetic are the paramedics that calm you down. So sympathetic means fight or flight. And this is the norepinephrine is responsible for that fight or flight sympathetic nervous system arousal. It's also responsible for hunger and alertness in our consciousness. Epinephrine is a transmitter that is pretty closely related to norepinephrine. It's also called adrenaline. It's located and produced in the pituitary gland, which is in our brain. And this is responsible for the fight part of fight or flight. And it can also be used to help manage allergic reactions. So, for example, in an EpiPen, it's epinephrine that is being injected, or adrenaline, which helps engage the sympathetic nervous system in a way so that it shuts down the allergic reaction that an individual is having. Serotonin is another neurotransmitter. This is a major player in terms of human experience, brain structure and function. Serotonin is located all over the brain. It is the major neurotransmitter that's involved in SSRIs, which stands for selective serotonin reuptake inhibitor, which are antidepressants. So what that means is SSRIs inhibit the reuptake of serotonin. So it stops extra serotonin from being reabsorbed by the axon of the first cell, which means it makes more serotonin available, which has a mood boosting effect on individuals with depression. It's also involved in so many other ways that we function, including the way that we perceive sensations in our sleep, our body temperature. This is a neurotransmitter that really runs the gamut of different human behaviors. Similarly, dopamine is a neurotransmitter that is a big part of our experience of pleasure and mood. But it does have concentrations within the brain, particularly the midbrain. So unlike serotonin, which is really kind of all over, dopamine has a large concentration in the midbrain and then some particular pathways that it tends to travel around within our brain. Many drugs affect this neurotransmitter. It is sort of the name of the game when it comes to understanding how drugs impact the brain. Your first guess will be dopamine. It's also linked to Parkinson's disease, as I mentioned in the first lecture, because of the concentration of dopamine in the substantia nigra in the midbrain, which is responsible for reward and movement. And so again, that Parkinson's disease, that disruption of movement, you're seeing dopamine being implicated there. Endorphins are a type of neurotransmitter that are located, again, in the pituitary gland and then also in the forebrain. And there's two different types of endorphins here. There are endogenous endorphins, which occur within the body. The body makes our own endorphins, and they are 100 times more powerful than morphine. So these are associated with our experience of pleasure. Endorphins stands for endogenous morphine, like endorphin. And so they're responsible for the sensation and the feeling of feeling good. And so again, those endogenous endorphins, the ones that occur within the body, super, super strong, super, super powerful. Then there are exogenous endorphins, which come from outside of the body, and those would be things like opiate drugs, like heroin. Those, again, are drugs that bind to the endorphin receptors that mimic the experience of pleasure that endogenous endorphins create. The seventh neurotransmitter we'll discuss is GABA. And this is both GABA and glutamate, the next two, are all over the brain. These are the major, what we call them, no-go and go neurotransmitters. So GABA, also called no-go, is the main inhibitory neurotransmitter. It stops the signal from continuing. And there are certain drugs that inhibit this neurotransmitter. And so if you inhibit something that's inhibitory, you get a double negative. So what happens is you stop the no-go signal from happening. And so you end up getting disinhibition and relaxation. So alcohol, Valium, these things kind of allow more free flowing of the neurotransmitter to occur. Glutamate is the go signal. It's the most abundant go neurotransmitter in the brain, and this gives the signal to keep sending the neural message, and it results in this excitation. And alcohol, PCP, and ketamine, but all three of those imitate glutamate in the brain. So there are many different ways that drugs can affect how neural transmission occurs, and it's all related to these neurotransmitters. So neurotransmitters are created within the neuron, so the cell makes these chemical messengers, and then they're packaged and put into the vesicles. And then those vesicles transport them down through the axon terminal, release them into the synaptic gap. The receptor sites take in what they can. It breaks down what they can't, or the original cell reuptakes the remaining neurotransmitter back into the cell. Drugs can affect neurotransmission at each one of these steps in the process. So moving from the synthesis and the creation of neurotransmitters all the way down to their reuptake into the cell. At any given point in that process, drugs can have an impact. So here are some examples of how that works. So the neurotransmitter synthesis, so the creation of neurotransmitters in the axon terminal of a neuron, drugs can increase or decrease the synthesis of neurotransmission or neurotransmitters. Transportation, so the movement of the neurotransmitter down the axon terminal through the vesicle, the drugs can interfere with the transport from cell body to vesicles. The storage in vesicles itself, so whether or not the vesicles can contain the neurotransmitter, drugs can impact that. So for example, they could cause the vesicles to leak neurotransmitter out. Drugs can be involved in the release of neurotransmitters, so it can cause a premature release or a release of more or less neurotransmitters than what would be without that drug. The breakdown of neurotransmitters within the synaptic gap, so drugs can increase or decrease that breakdown by the enzymes. The reuptake, so drugs can stop the reuptake of extra neurotransmitters in the synaptic gap. It can keep those neurotransmitters in the gap longer. And on the other side, of the receptor side, the dendrite side, it can also have an impact on those receptors. So drugs can either mimic a neurotransmitter, as in this case, and activate the receptor site. So this would be an agonist. So For example, methadone is a drug that binds to the endogenous endorphin receptor sites. And so it then activates those receptor sites and prevents other drugs from activating it. That would be an agonist. Or it could block a receptor site. So it can occupy a neurotransmitter receptor, bind with that receptor, and prevent it from activating at all. And so this would be an antagonist. And an example of that is naltrexone, which is in Narcan. So whereas methadone activates a receptor site, binds with the same receptor site as heroin, for example, and activates it, naltrexone or Narcan activates to that same attaches to that same receptor site, but blocks it from being transmitted. So it actually stops the experience of what heroin would be doing to the neurons. So that's it for brain basics. And we will, in our next lesson, be talking about more particular the basics of drugs. So how do drugs get into the body? What do they do once they're there? How do we understand that? But with this understanding of our basic functioning of the brain and the nervous system, we can then start to understand, okay, what happens when this is disrupted through the introduction of some sort of substance.